Our next speaker is Dr. Lindsey MUIR from University of Michigan. Lindsey's background is in molecular and cell biology, and her PhD is in cell biology from University of Washington. And her main area is obesity and immune-related reprogramming. So her talk today is Genomics of Human Adipose Tissue Macrophages in Obesity. Thank you, Lindsey.
Thank you for inviting me to speak today. It's an honor to be here with you all and with some great academics here. So in my work, I am very interested in how we can use genomics data from human cells to understand the immune system. And from there, use those data to understand how we can reprogram the immune system for health benefit, and also to combat disease and lasting damage from injury. So in my talk today, I'm going to focus on how cells in the immune system, specifically cells called macrophages, change with obesity. And then I'll touch a little bit on our thinking in terms of how we might reprogram immune cells. So before I give some of the genomics data on immune cells, I want to introduce a bit about obesity and what we know so far. So we know that obesity is a top global social burden, costing about US $2 trillion in 2012. Obesity is also increasing globally, and as are healthcare costs. So in the future, this may be even more of a social burden. To give you an idea of the current state of obesity, I have a few statistics to share looking at Hong Kong, China, and compared to the US. And so obesity is usually measured by body mass index, or BMI, so just kilograms per meter squared. And keep in mind that in, I think, Hong Kong and China, BMI of 25 or more is considered obese, whereas in the US it's 30 or more. So in Hong Kong, obesity is about 20%, with about 1.5 million people, and it costs about 5 billion Hong Kong dollars per year. This cost is from obesity-related conditions like type 2 diabetes and heart disease. China has a much lower frequency of obesity, but still accounts for a very large number of people who are obese. And in particular, the number of children who are obese is increasing a lot. And so in the future, this may become a much bigger problem. So looking at the United States, we are approaching 40% obesity, which is a massive number, costing an estimated over 300 billion per year at this point. And it's costing about 20% of our healthcare costs. And this includes, like I said, heart disease and metabolic syndrome, diabetes, all of these related conditions. So we know that obesity is a huge risk factor for diabetes, heart disease, metabolic syndrome, and also these secondary conditions such as asthma, sleep apnea, cancer, and it affects the risk for all of these. So the longer you have obesity, the idea is that you may be at risk as that goes on in a chronic obesity situation for many, many medical conditions. As we know, prevention is the #1 way that we can address this issue, but there are interventions that can improve health. So we can manage comorbid disease as well as help promote weight loss. One of the ways to promote weight loss is medical or behavioral, where you can work with a physician to help reduce the weight and help manage the conditions associated with obesity. But unfortunately, the response of diabetes to this intervention is about 5%. So 5% of the people who undergo this medical weight loss actually have remission of diabetes. More effective is surgical weight loss, things like bariatric surgery, where they can remove part of the stomach and intestines, or put a band around the stomach to help reduce nutrient intake. This is effective in about 50% of the people who undergo this procedure. However, in about 50%, they don't see any benefit. And one of the big problems is that we can't distinguish between the patients who will benefit from this and those who won't. So it really will help us if we can understand more about obesity, how it affects cell and tissue function, and find new ways to combat this. So another thing to mention, as I mentioned before, in China in particular, obesity in children is becoming a lot more common. And we know that in lean adults, so adults who are normal weight but had childhood obesity, are still at much higher risk of disease. So even when they lose the weight, they still have a high risk of disease. Okay, so what do we know about obesity here? What happens to us in obesity? With overnutrition, our adipose tissue, or our fat tissue, expands massively, and this tissue itself then can become insulin resistant, so it can have difficulty storing energy after that. This leads to metabolic problems, including whole body insulin resistance and diabetes and metabolic syndrome. So interestingly, immune cell activity that increases inflammation has been strongly associated with fat tissue expansion and development of diabetes in mice. And this connection is a little bit less strong in humans because we don't have a whole lot of data on it. So I want to show you a cross-section of adipose tissue so you'll be familiar with the cells that I will be talking about. And we can see adipocytes here in blue, as well as blood vessels shown here, and all cell nuclei here are shown in red. So you can see the blood vessel cell nuclei. And then in green here we can see macrophages, and we call these ATMs for adipose tissue macrophages. So these in mice have been associated with diabetes, development of diabetes and obesity. So this hasn't been well defined in humans, so we wanted to get a better grasp of this in human adipose tissue. So we worked with a collaborator, Robert O'Rourke, to collect adipose tissue from human patients from our bariatric surgery clinic. And he is one of the bariatric surgeons who also runs a lab studying adipose tissue. So we mainly study adults 21 to 60 years old with a BMI of greater than 30. So I won't go through this, but just to show you that our main comparison here is between obese patients who have diabetes and those who don't have diabetes. And it was a pretty good mix of male and female, a variety of ages. And also, just for comparison, a lean population. So one of the first things we did was to look at whole tissue gene expression. So we take adipose tissue and collect all expressed genes, all RNA, and do RNA sequencing. And from that we can get quantify the total expression of all genes in the tissue. And looking here at the most dominant features of abdominal adipose tissue compared to subcutaneous adipose tissue, we can see that our diabetic patients have a much different signature compared to our non-diabetic patients in terms of the whole tissue gene expression. So similar in subcutaneous here, diabetic is much different from non-diabetic gene expression. So when we do a pathway analysis, so this just tells us the cellular pathways that are dominant in the gene expression signature. And you can see that in visceral abdominal adipose tissue, our diabetic patients have an inflammatory signature. This comes up again and again. So there are immune cells here that are doing something. They're doing something different in the diabetic patients. And we don't know whether that's causal yet, but we're trying to understand more about this. So in mice, it's known that inflammation is associated with macrophages in particular. And much of what we know about ATMs, because it's studied in mice, we wanted to approach this in humans. So we can see just a cartoon of adipose tissue macrophages here, and they increase massively in obesity as adipocyte volume increases with energy storage. So these cells seem to keep pace as the tissue expands. They keep pace with that expansion, and you retain a certain quantity within that tissue. So you can imagine that if that tissue gets very large, you have a massive increase in the number in your whole body, the number of macrophages present. So because of these studies were done a lot in mice, when I started this work, we didn't have a good idea about what proteins, how do we define these cells? How do we look at them? And what are the markers, the protein markers on their surface that we can use to identify them? So for these studies, I collected human adipose tissue, and you digest it enzymatically so you can get a single cell suspension. And then you can isolate everything that's not an adipocyte, which we call stromal cells or stromal vascular cells. We can then use flow cytometry, adding antibodies that detect specific surface proteins, and in that way identify the different cell types and the different macrophages that are there. So we usually look at this data on a two-dimensional plot. So here is one of our markers for macrophages, CD64 on this axis, and on this axis CD45. So that's just a marker for immune cells. And so by looking at the data this way, we can see that our macrophages lie right up here. We can then take only the data from these cells and further divide them into 3 different cell types, which we didn't know when we started what we would find, but these markers in particular seem to be good at identifying different macrophage types. So we have 3 types, 206 positive, DP4 double positive cells, and then CD11C positive. So based on fluorescence intensity on these axes. So using flow cytometry, we can quantify these 3 types of ATMs in different patient populations and see how they're different in patients with disease compared to those without disease. And a primary finding here is that when we quantify CD206 positive macrophages, so this is just a frequency in the total population, they're increased in obese diabetic patients compared to obese non-diabetic patients and compared to lean patients here. When we look at the double positive cells, these are also increased in our obese diabetic patients compared to non-diabetic and compared to lean patients. But when we look at this type of macrophages, we don't see the same increase. They seem to be very stable across these populations. Again, in humans, high variability with our patient population, which is typical. Okay, so we wanted to understand more about their function of each of these ATM subtypes. So we took each population and flow sorted them for RNA sequencing so that we could understand their whole gene expression signatures. When we compare them, we see that the 206 positive differ from both of the other subtypes, so they seem to diverge in terms of their gene expression from both other subtypes. So looking at these comparisons, total of 831 differentially expressed genes, so significant difference in these genes in terms of their expression between 206 positive and 11C positive, and then between 206 positive compared to the double positive. Many genes are different. However, comparing these 2 populations here, we can see that it's a much lower number of genes that are differently expressed. So using principal component analysis, we can also see that the signature of 206 positive are much different. So up here compared to double positive or 11C positive. And when we put this, use a dendrogram analysis, we can see that these 2 are cluster together compared to 206 positive, which diverge. So all of this just to say that these seem to be divergent in terms of their gene expression. We still don't know much about their function. And can we use data to really understand more about their function? Okay, so using biological pathways, we can begin to see signatures within each of these cell types, each of these macrophage types. So in these columns, it's just showing the comparison of 206 to each of the other populations, and I didn't show the comparison between these two as they weren't as much different. So we identified more than 20 different pathways that were significantly different in 206 positive ATMs compared to both other subtypes. I'm just showing a subset of those here. So we can see that significant pathways that were different relate to cytokines, which are important signaling molecules, cell metabolism, as well as internalization. And macrophages themselves, they're professional phagocytes, which means they eat and they digest parts of the tissue, and they're heavily involved in tissue repair. So one of their functions is to detect and sense their environment and then internalize particles from the environment, whether that's a foreign pathogen or part of the tissue itself that needs to be recycled. So it's not surprising that they would have genes related to cell internalization, that they take in environmental signals. But the 206 positives seem to diverge greatly in terms of that function compared to the other two subtypes. So to dig a little deeper into their potential functions, we observed that many of these significant pathways related to internalization. So scavenger receptors are related to that, so they're cell surface receptors that detect patterns in the environment and then facilitate internalization of different particles. Endocytosis, so just taking in of environmental particles, and then also phagosomes. So all of these pathways came up as significant in the 206 positive, and more genes are expressed in the 206 positive here compared to the 11C positive ATMs. So we thought that internalization might be a significant function of these cells compared to the other two types, as well as this internalization is also highly related to antigen presentation. So these cells take up particles and they digest them and then present to other immune cells to stimulate their function. And so we thought that this may be a prominent function of these cells. And indeed, when we look at an antigen presentation surface protein, HLA-DR, we can see that the cells that express high HLA-DR are majority 206 positive on this axis. Okay, so one thing that we couldn't answer with this, why are 206 positive increase? So we see this increase in diabetic patients compared to non. There are many different cues in the environment of obesity. So as the tissue expands, you get more free fatty acids, more lipoproteins, glucocorticoids, lots of molecular signaling going on. But we don't know yet what is it that's stimulating these cells to increase. So with this increase, we think these cells are detecting environmental cues and are becoming more sensitive to environmental cues and performing more antigen presentation and are heavily involved in inflammation in this way. So this can lead to tissue dysfunction as well as development of diabetes and insulin resistance. So another important question about macrophages is where are they coming from? And traditionally, they're thought to come from circulating cells called monocytes. And so I want to next discuss some of my more recent work on human monocytes and obesity and how we may be able to reprogram them to behave differently in disease, in conditions like obesity, but also other diseases that have an inflammatory component. So monocytes are interesting because they find sites of tissue damage, so they circulate throughout the body, and then at different times they can home to sites of inflammation or injury. And so that's an advantage of targeting monocytes, is that they can find tissue damage, and then they will differentiate or become macrophages within the tissue. So there's great potential for these to modify these responses and thus control inflammation. So again, using flow cytometry, we can identify different types of monocytes by their expression of surface proteins. And two of these surface proteins have been used to identify human monocytes, and these are CD16 and CD14. And these distinguish three types of monocytes in humans. NC, this is non-classical. INT stands for intermediate, and then classical monocytes here. So classical, high in CD14 and low in CD16, and inverted for non-classical. So these monocytes are known to increase in obesity, but in terms of understanding how they behave in humans, not much is known. And we wanted to not only more deeply understand these, but also figure out different ways to reprogram them. So we were working with Ronnie Shea in the University of Michigan Department of Biomedical Engineering. So he is using different types of polymer nanoparticles and studying them in the context of mice, so injury models, and to understand how we can modify their response in injury. So they showed that these interact with mouse monocytes through scavenger receptors. And if you remember, those are the receptors on the surface that interact with environmental signals and can help in internalization. So it makes sense that they would interact with these receptors. And mouse monocytes then interact with the nanoparticles through these receptors and internalize them. And we're working with two nanoparticle types in particular and modifying their size as well as their charge to see how they interact with human monocytes. So PLA, or polylactic acid, and PLG, polylactide coglycolide. So these two nanoparticle types. So our goal was then to test their interaction in human monocytes from patients who are obese. So these nanoparticles, I'll also mention, were conjugated to a fluorescent particle, Cy5.5, and in this way we were able to detect interactions with cells through the cells being positive for that fluorescence. So when we apply these nanoparticles to monocytes, we can then evaluate them using flow cytometry and quantify which types become positive for that nanoparticle fluorescence. And we can see here, so this is showing non-classical monocytes, and then when we apply nanoparticles, we can see that they do interact. So this axis is just showing the nanoparticle fluorescence, so high fluorescence and low fluorescence. Without nanoparticles, the signal is here. When we add nanoparticles, we can see that the non-classical interact somewhat. Even more, though, the classical monocytes interact with these nanoparticles. And I should say this makes sense because classical monocytes actually have higher expression of scavenger receptors. And in fact, we also know that in diabetic patients, when we look at the whole tissue gene expression, we can see an increase in scavenger receptor expression. So that's an important feature that we may be able to use. And when we add these nanoparticles to monocytes from either obese patients or obese patients with diabetes, we can see that the obese patients with diabetes actually have monocytes that interact more with these nanoparticles. So this is evidence toward we may be able to specifically target monocytes that are active in disease and maybe facilitating tissue damage so that we can modify their inflammatory properties. So one of the directions we want to take this is to genomics characterization of human monocytes. So we have done tissue macrophages, so they're offspring, I guess, of monocytes, and we want to look at these human monocytes in obesity and do a genomics characterization. So our goal is to do RNA sequencing for whole gene expression in these cells and these different monocyte types, as well as Hi-C, so being understanding how genome conformation is different within each of these cell types. And then we want to identify from the data, distinct functional phenotypes looking specifically at transcription factors. So we know that these can control cell behavior and cell fate and cell types. So we want to look at transcription factors to see what the subsets are that are active in certain types of monocytes. And then using that information, we can potentially modulate those types using nanoparticles that can target specific monocytes, and also to modify their expression of specific transcription factors. So one of the bottom lines here, obviously, is that we want to prevent chronic obesity because we know that there's a higher risk of disease, cardiovascular disease, diabetes, metabolic syndrome. There's a sustained disease risk. So we know that childhood obesity can lead to, even when you're lean as an adult, can lead to higher risk for disease. And we know that we can only induce remission in about half of the bariatric surgery patients. So we do have an effective method, but it's only effective for about half the people who undergo the procedure. So we know that from these human and mouse studies that obesity can permanently damage tissues such as adipose tissue. And we know in data that I didn't show today that inflammation does not resolve with weight loss. And we've done a number of studies in mice that show that those inflammatory cells actually stick around, they stay. Even when the tissue contracts and you have weight loss, they don't resolve and they remain in the tissue. So we're not sure. We're hoping that would resolve eventually, but we haven't taken the studies out long enough to understand when that resolves, if it does. So the idea then would be to reprogram immune cells into a potentially that obesity can reprogram them into potentially harmful phenotype or a metabolic phenotype. And can we take it back? Can we reprogram those into a beneficial type that will promote metabolic health? So the other focus of my talk was using data to understand function and programming of human immune cells. And we know that in terms of ATMs, the macrophages, the 206 positive macrophages are a distinct type that is associated with metabolic disease, and that they seem to have a primary function in environment detection, internalization, antigen presentation. So all the genomic signatures pointed to that as a potential function. We know that monocytes have a distinct response to obesity, and that they interact differently with nanoparticles in metabolic disease. So our diabetic patients seem to interact with them more, suggesting a different complement of surface proteins that mediate that interaction. And I'd like to conclude by acknowledging most of this work was done in the lab of Carrie Lu Meng in the Department of Pediatrics at the University of Michigan. I've since moved to computational medicine at University of Michigan. Our collaborator, Robert O'Rourke, as well as indica lagipoxa, which I hope to do more work on the genomics and mathematics as well with indica. Affiliated labs, our bioinformatics team, and of course our funding sources. Thank you. I'll take any questions. Yeah.
For this important problem, costing $300 billion for obesity. So how you think how among these government, insurance company, or people themselves who have the most incentive to solve this problem? You think?
Yeah, and I think with the different healthcare systems, it may have a different burden on the government compared to people paying into the system. But I think certainly everyone, because I think as obesity increases, the cost for everyone, I think in most cases, people pay a little bit in. But if you have higher health burden, then everyone is going to pay in way more than, much more than if that burden were lower. And I think especially considering the increase in childhood obesity, that will be a significant problem in 10 to 20 years, as well as increase in healthcare costs happening during that time. So I think it would benefit greatly to try to address these issues.
That's right, but you think who have the incentive to, because now it's eventually paid by all the people, but now to solve this problem, we need to find some key players who would like to pay to solve this problem. Like government,
Yeah, absolutely. And I think government should play a role in this, in trying to implement and also fund studies on what is the best way to intervene, especially in children and potentially in schools, to try to promote education and to help people I guess manage this problem and know the best nutrition. So certainly governments can play a role, and I think there are already programs and interest in doing this globally.
I can make one comment. Now Amazon is so much into human health. Amazon. Amazon.
Yes, Amazon.
Yeah, I think there's been a lot of interest. Amazon and Nike even has come in and they want to do more programs. Yeah.
And you showed different uptake of nanoparticles by different subsets of monocytes in patients with diabetes associated obesity. So are you postulating that these monocytes are already abnormal before they infiltrate into the fat?
I think they are.
Means that it's not the fat that turn them into abnormal.
I think it's the environment of obesity. There's something there, and we haven't pinpointed what it is, only indirectly through the cells themselves and the surface protein expressed in those cells and the gene expression profiles. But I think we can trace back to see potentially which surface receptors are differentially expressed and explore how those modify cell function, because I think this continual environment of free fatty acids, different nutritional components actually stimulate them a lot. These cells are incredibly sensitive to their environment, so prolonged stimulation through specific diets would surely affect them. Yeah. But then I also add, as they infiltrate the tissue and differentiate, we also think that process is highly affected. So if they, the starting point is a very different expression profile, then when they differentiate, that should also be different. So within the tissue environment, you will have input by the tissue, free fatty acids or whatever the adipocyte signaling is that's different in obesity as well. Mm-hmm.
So Lindsey, this is a very nice set of studies and a huge problem. In fact, in America, been documented for the last 40 years. Percentage of adults, the percentage of adolescents, children, over the barrier, the threshold for obese or overweight has been growing rapidly, and there's been no amelioration by people. One of the problems in biomedical research is that it's very useful to study animal models, mouse models, to explore possible mechanisms. But there's always the question whether the mouse model has anything informative for the human. That's particularly a problem when the mouse model may not even predict for other mice. So educate us, please, about where you think the mouse or models you're studying are most likely to be informative and transferable, and where you've discovered that they may not.
Right, so I think where they're useful is in terms of understanding systemic response, so whole body response, circulating cells, these things, and as well as understanding the lineages of cells that are involved. So things like lineage tracing, which we can't do well in humans yet, is where that will be very useful. What was yours? You had a second question?
Is there a parallel to bariatric surgery in the mouse?
There is, yeah.
Only 50% of them respond?
I don't know that. Yeah, I don't know that. Certainly at the tissue level and the cell level, the markers that identify immune cells have been found to be very different. So I think these kinds of studies where we actually have cohorts of human patients are extremely important. And we weren't sure when we started. There were very few papers published on what markers were there, what to use, and so we struggled a bit in the beginning to find a good set of markers. And we still don't know. I think there's a lot to be done here, but it certainly wasn't the same as the mouse cells. So.
Yeah, I think Dr. Oman, you touching the more essence here is that whether the mouse models, how that reflects human system. I always had this question myself too.
Yeah.
And maybe some sort of general functions like circadian maybe reflect in animal or human similarities. But that's a big question whether the like obesity or cancer. I don't know how that reflects in animal model versus human. That's where maybe the reprogramming might help.
Next question. Yes. You mentioned reprogramming in the model and in the humans, as well as your own interest in reprogramming for therapeutic trials. But understanding that reprogramming is part of the disease process should be important.
Absolutely, and I think this is where to understand between lean and obese, but also between obese, the people who actually get diabetes, and some obese people will never get diabetes or a metabolic problem. So to understand those differences in terms of their gene expression signatures, to understand what is different about those will be very important.
I think we have enough time. Any more
Any other
questions?
I know this is not a biology audience, so if you have any basic questions too, I'm happy to answer.
You want?
Yeah, I have a very stupid question. You mentioned in the middle of the inflammation cells when people lose weight will not disappear, still there. So does that suggest the immune system have to be adapted to clean those cells? That's not so easily reprogrammable.
And I think, so this is where we
The
had,
question.
yeah.
Make sure everybody understands the question.
Oh, yeah, so how, can you restate what you said?
I just wonder how the cells with inflammation cannot be that maybe exists
To resolve.
Yeah, after people losing the weight.
Right, so.
How the immune system can.
Right. So I had mentioned that the immune cells within adipose didn't go away after weight loss. So this is something we don't know much about yet, because we don't know sort of the timing of when this might resolve. And certainly there's a component of tissue damage where we see, and even in the human we did some studies on fibrosis. So the matrix that holds all the cells, right? So that changes dramatically as well. And so all of these things, I think, contribute to these cells staying within the tissue with weight loss. And there's a lot of tissue repair to happen to resolve and to reconstruct what the tissue was before. But we don't know whether it will ever go back. And this may be one reason why with obesity you have, for all of us, it's difficult. So once you reach a state of obesity, it may be very difficult to go back. So there may be some permanent changes that happen if you go long enough and have a chronic obesity situation. Maybe 1 to 2 years, okay, you can lose weight and resolve completely. But longer than that, we don't know the time scales, certainly. But I think the immune system definitely has a function in repairing the tissue and resolving the tissue damage that can happen.
That would make childhood obesity all the more damaging.
Absolutely. So consider when this happens in adulthood, we see this, but then in childhood obesity, the long-term effects can be pretty serious.
A question about genetic inheritance of risk for obesity, and specifically what that suggests about educating parents who may be obese or maybe were obese as children about what they should be doing about their own children in a more urgent sense. If you're talking about priority populations for intervention, I'd be interested in your thoughts.
And certainly genetics should play a role. It's difficult, obviously, to separate the genetics from, the cultural and family culture behind eating. But I think that will be a huge key to modifying behaviors. Is number one, education and targeting the parents who need it the most. And it tends to be associated with socioeconomic status and education level. But I think certainly for all of us, as we know, food is good, and we love food, and we love to eat many times and eat food that tastes good. And so I think it's going to be many factors that we have to address. But I think certainly targeting families and parents, and in particular, will be very important.
Are there genetic differences between those adults who have the diabetes problem in addition as opposed to those not?
That I don't know. Yeah. So I think we only looked at, in my data, looked at gene expression. So not sort of the genetic backbone of what variation was there, but certainly that could play a role. Mm-hmm.
So I really like the Yuan's question. So that means there's some sort of memory, right? Like a cellular memory, how that is affected by obesity. Is there evidence for?
Yeah, and I think that certainly can be playing a role. Yeah, so I think that because these cells are so tuned to their environment and sensing their environment, they are programmed by that to behave differently. So when we're talking about monocytes, they circulate in the body for a few days or a week, but then they're replaced by new monocytes. Going back to the origin of those monocytes, in mice we see that the bone marrow actually changes as well in obesity. So when they're exposed to obesity, the cells in the bone marrow become the blood cells, and those cells then are different from how they were when they were not exposed to obesity. So I think in that sense, it should happen from the progenitor all the way to the differentiated cell, and some of those cells stick around for longer than others. So when we consider an adipose tissue macrophage, those are relatively long-lived compared to monocytes. So if they infiltrate a tissue and differentiate in the obesity environment, those could stay in the tissue for much longer and promote inflammation and dysfunction within the tissue for a longer time. Okay.
Any other-
Stephen?
Yeah, as far as immune response, damage to cells, etc., do we see, like, is there a sliding scale between being overweight, obese, morbidly obese, or is there kind of a threshold where you start to see these kind of gene expression differences and other problems arise?
I'd say that's really unknown in humans. So in my studies of mice, we had looked at short-term diets also. So in mice, when you induce obesity, you can do it genetically, or you can feed the mice a high-fat diet, and they will get obese, and their fat pads will expand dramatically. And I've done series of high-fat diets over different times, and the fat pads expand quite rapidly in the beginning, but then they reach sort of a stable quantity or a stable volume in the tissue. And sometime between when they form the maximal size and a number of weeks later on this diet, there's a time window where we see massive increase in inflammatory macrophages. So the problem really is with the humans, we don't know what that time window is. And I think human diets are obviously much more variable than, the control diet for the mice. But I think you're right that there is sort of a time window of how do we define chronic obesity. But there may be a threshold there where if you pass it, it becomes much more difficult to lose weight and to restore the lean physiology. Okay.
Thank you very much. So we have another session tomorrow at 2 and 3:30. Please come to tomorrow's sessions too. So I think we are done for today. Thank you so much for coming.